This is a creative recreation using artificial intelligence. The voice and ideas presented here are inspired by the life, writings, and documented scientific work of Albert Einstein. This is not a real recording or statement from Einstein himself. All scientific content in this presentation is based on documented published research, peer-reviewed studies, and verified historical record. Where research is contested or disputed, it is presented as such. This channel is committed to scientific accuracy. Nothing presented here should be interpreted as medical advice. There is something you were never taught in school about sound, not because it was hidden from you deliberately, but because what sound actually does to living matter is so unsettling that the scientific establishment spent decades refusing to look at it directly. Sound is not simply vibration detected by the ear. Sound is mechanical energy propagating through matter. It compresses and rarefies the medium through which it travels. At sufficient intensity and specific frequencies, sound physically reorganizes objects. This is not philosophy. This is documented physics with a 200-year experimental record. In 1787, a German physicist named Ernst Florenz Friedrich Chladni placed sand on metal plates and drew a violin bow across the edge. The sand did not scatter randomly. It moved. It organized itself into precise geometric patterns, symmetrical, intricate, repeating. Different frequencies produced different patterns. The same plate, the same sand, a different frequency, an entirely different geometry emerged. Chladni's figures, as they came to be called, demonstrated something that most people still do not fully appreciate. Sound does not just pass through matter. Sound organizes matter. Sound imposes structure. In the 20th century, researchers began asking the question that Chladni's plates made inevitable. If sound organizes grains of sand into geometric patterns on a metal plate, what does it do to something far more complex? What does it do to a living cell? What does it do to the molecule at the center of all life, deoxyribonucleic acid, the double helix that carries the instructions for building and operating every living organism on Earth? The answer that emerged from decades of research was more disturbing than the question. Not because the answer was dark, but because the answer was real, and real things have consequences that cannot be easily managed or dismissed. I have spent my life thinking about the nature of energy and the nature of matter, and what I came to understand, what the equations forced me to understand, is that the boundary between a wave and a particle is not absolute. The boundary between vibration and matter is a matter of scale and perspective. Everything that we call solid, everything we call physical and fixed, is at its foundation a pattern of energy in motion. The electron that orbits the nucleus of an atom is not simply a particle sitting in place. It is a wave function. It is probability distributed through space. When you understand this, the idea that sound, organized mechanical vibration, can interact with biological molecules in profound and specific ways does not seem strange at all. It seems inevitable. My name is Albert Einstein. I was born in Ulm, in the Kingdom of Württemberg, on the 14th of March, 1879. I spent my career at the Patent Office in Bern, at the University of Zurich, at the Kaiser Wilhelm Institute in Berlin, and finally at the Institute for Advanced Study in Princeton. I am remembered for the theory of relativity and for the equation E equals mc squared. But the work I found most important was the work on the quantum nature of light, the photoelectric effect, which demonstrated that light, which everyone understood as a wave, also behaves as a particle. That discovery changed everything. If you find what I am about to tell you interesting, I encourage you to subscribe to this channel. More questions like this one are being explored here. Questions that the textbooks have not yet caught up with. Let us begin with what is not disputed. Medicine already uses sound to affect biological tissue in ways that would have seemed like magic 2 centuries ago. Ultrasound imaging uses frequencies between 1 and 18 megahertz to create images of a fetus inside the womb. The sound waves penetrate tissue, reflect off structures of different density, and return information that a computer assembles into an image. This is mechanical vibration doing work inside a living human body. No one disputes this. It is standard medical practice performed millions of times each day around the world. More remarkable still is a procedure called lithotripsy. A patient with kidney stones is positioned near a device that generates focused ultrasonic pulses. The sound waves are directed at the kidney stone with precision, and the stone, a hard calcified mass inside a living human body, shatters. Not because a surgeon cut it, not because a drug dissolved it, because focused sound at the right frequency delivered sufficient mechanical energy to break the stone apart. The body then passes the fragments naturally. This is approved by the United States Food and Drug Administration. It is performed in hospitals. It is not alternative medicine. It is the documented mechanical power of sound operating inside biological tissue. Ultrasound also accelerates healing. Low-intensity pulsed ultrasound, known in clinical literature as LIPUS, has been shown in peer-reviewed studies to accelerate bone fracture healing. The mechanical vibration stimulates osteoblasts, bone-forming cells, to increase their activity. A 1994 study published in the Journal of Bone and Joint Surgery demonstrated a 24% reduction in healing time for fresh fractures treated with LIPUS compared to controls. The FDA cleared this treatment. It works. The mechanism is called acoustic mechanotransduction, the process by which cells convert mechanical vibration into biochemical signals that alter cellular behavior. This is the concept that matters most for what we are about to discuss. Acoustic mechanotransduction. Cells are not passive recipients of their environment. They are active sensing systems. The membrane of a cell contains proteins called mechanoreceptors, structures that respond to physical force. When mechanical vibration reaches a cell at the right frequency and amplitude, those receptors open. Ion channels shift, signaling cascades begin, gene expression changes. This is not theoretical. This process has been documented in the peer-reviewed literature in studies of cardiac cells, bone cells, stem cells, and neuronal cells. Sound, mechanical vibration, literally changes which genes inside a cell are expressed at a given moment. The resonant frequency of a cell depends on its size, its membrane composition, and its internal structure. Red blood cells have a different resonant frequency than liver cells. Cancer cells, which have altered membrane properties and different cytoskeletal organization, have different resonant frequencies than the healthy cells from which they derive. This is not a fringe claim. It is the basis of a growing field called mechanobiology, the study of how mechanical forces regulate biological function. The journal Nature published a review of mechanobiology research in 2014, noting that mechanical signals are as important to cell function as chemical signals. Sound is a mechanical signal. Now I want to connect this to something I spent years thinking about: the quantum nature of energy. In 1905, I was working at the patent office in Bern when I published the paper on the photoelectric effect. The question I was addressing seems simple. Why does shining light on certain metals produce an electric current? The classical wave theory of light predicted that any frequency of light, given sufficient intensity, should be able to eject electrons from the metal surface. But that was not what the experiments showed. Only light above a certain frequency, regardless of intensity, could eject electrons. Below that threshold, no amount of brightness worked. Above it, even dim light ejected electrons immediately. The explanation required accepting that light does not travel as a continuous wave. Light travels in discrete packets, quanta, which I called photons. Each photon carries an amount of energy proportional to its frequency. A low-frequency photon does not have enough energy to eject an electron, no matter how many photons arrive. A high-frequency photon has enough energy to do the work in a single interaction. This won me the Nobel Prize in Physics in 1921. But what I want you to notice is the underlying principle. Frequency determines energy. Frequency determines what interactions are possible. Different frequencies do fundamentally different things. Sound, at the quantum scale, also travels in discrete packets. Physicists call them phonons. A phonon is a quantum of acoustic energy, a quantized mode of vibration in a crystal lattice or other material structure. Phonons carry energy and momentum. They interact with electrons. In condensed metaphysics, the study of phonon behavior is essential to understanding superconductivity, thermal conductivity, and the behavior of solid materials at low temperatures. At the scale of biological molecules, vibrational modes, which are phonon modes in a quantum mechanical description, govern molecular flexibility and function. The DNA molecule, for example, has documented vibrational modes that affect how it bends, how it opens for transcription, and how proteins interact with it. A 2016 paper published in the journal PLOS ONE by researchers at the University of Buckingham examined the effect of specific sound frequencies on DNA using Raman spectroscopy, a technique that measures molecular vibration. The researchers found measurable changes in the vibrational signature of DNA samples exposed to specific audio frequencies. This was a small study with limitations the authors acknowledged, but the measurement was real. The effect was measurable. The question was not settled, it was opened. That is how science is supposed to work. The principle that connects all of this, that frequency determines the nature of physical interaction, that waves and matter are not as separate as they appear, that the universe at its foundation is a structure of vibration, is not mysticism. It is the conclusion that the mathematics of quantum mechanics and the evidence of a century of experiments have forced upon us. The famous inventor Nikola Tesla once articulated a version of this idea in his own terms. "I will not attribute words to myself that were not mine." But the principle that energy, frequency, and vibration are the fundamental language of the physical world is consistent with everything I derived mathematically. Now I want to tell you about the research that the established medical community did not want to take seriously. In the 1930s, an American physician and inventor named Royal Raymond Rife developed a microscope of extraordinary magnification and resolution that he claimed allowed him to observe living microorganisms in real time at magnifications far beyond what conventional optical microscopes of the era could achieve. Using this microscope, Rife identified what he called the mortal oscillatory rate of various microorganisms, the specific frequency at which, he claimed, each organism would be destroyed by resonant vibration, much as a wine glass shatters when a singer hits the right note. Rife's claims were dramatic. He reported being able to devitalize cancer-causing microorganisms using targeted frequency radiation. In 1934, a clinical study was conducted at the University of Southern California involving 16 cancer patients. The researchers reported complete remission in all 16 patients after 90 days of treatment with Rife's frequency device. This is what Rife's supporters document. What is also documented is this: the study was never properly replicated in controlled conditions. Rife's claims about the nature of cancer, specifically that a filterable virus was the universal cause, were not consistent with what subsequent research established about cancer biology. The science was contested, and the contestation was legitimate. What happened next is where the institutional history becomes genuinely troubling. The American Medical Association, under the leadership of Maurice Fishbein, aggressively pursued legal and professional action against practitioners using Rife's device. Rife's collaborator, Dr. Arthur Kendall, faced severe professional pressure. The laboratory at 3901 Olcott Street in San Diego was broken into. Research materials were destroyed. This is documented in historical accounts of the period, though the full picture is disputed. What is not disputed is that the medical establishment of the mid-20th century had structural reasons, financial and professional, to resist therapeutic approaches that did not involve pharmaceutical products. This is not conspiracy. This is institutional sociology. Institutions protect their paradigms. The NASA experiment with sound and biology is more recent and less contested. In a study examining plant growth in microgravity environments, NASA researchers documented that specific acoustic frequencies, particularly in the range of 100 to 1000 Hz, influenced the germination rate and root development of plants. The research was conducted in the context of developing food production systems for long-duration space missions. It was practical agricultural research. The finding that sound affected plant biology was a secondary result that the researchers themselves noted warranted further investigation. More recent work in agricultural acoustics has built on this, with studies published in peer-reviewed journals documenting measurable effects of acoustic treatment on seed germination and crop yield. The word band in the context of bioacoustic research requires precision. No government agency has issued a formal prohibition on sound frequency research. What happened, and continues to happen, is more subtle. Funding agencies, editorial boards, and tenure committees are composed of human beings with professional investments in existing frameworks. Research that challenges established models faces a higher burden of proof than research that confirms them. This is not unique to bioacoustics. It has characterized every paradigm shift in the history of science. I know this personally. When I submitted the paper on the photoelectric effect in 1905, the concept of quantized light was so contrary to the prevailing understanding that it took 16 years and the Nobel Committee to validate it. I once wrote that great spirits have always encountered violent opposition from mediocre minds. I do not say this to be cruel. I say it because it is observably true, and because the history of science is not a smooth progression. It is a series of struggles between what the evidence demands and what the established framework can accommodate. The question of how sound frequencies interact with DNA, with the molecule that carries life's instructions, is a question that the evidence is demanding we take seriously. The documented mechanisms of acoustic mechanotransduction, the measurable effects of ultrasound on cellular gene expression, the phonon physics of DNA vibration modes, these are not fringe ideas. They are threads of a picture that is beginning to resolve into something coherent. What does this mean for you? I want to be precise here because precision matters in science and also in how people act on scientific information. The documented evidence tells us several things clearly. Sound at sufficient intensity and appropriate frequency causes measurable biological effects. Cells convert mechanical vibration into biochemical signals. Those signals affect gene expression. Those effects vary by frequency, amplitude, duration of exposure, and cell type. This is established in the peer-reviewed literature. What the evidence does not yet clearly establish is what specific frequencies, at what specific intensities, produce what specific therapeutic effects in human beings under controlled conditions. The research is real, but incomplete. The mechanisms are documented, but the dose-response relationships in clinical human applications are not. This is where more research is needed. The barrier to that research is not technical. The technical tools, precise frequency generators, advanced genomic sequencing, real-time cellular imaging, are more capable now than at any point in history. The barrier is institutional willingness to fund research that does not fit the pharmaceutical model of intervention. My own experience of the universe has always been one of deep conviction that what we do not yet understand is more interesting than what we do. The fact that I spent years deriving equations that described the nature of space and time did not diminish my sense of wonder. It deepened it. Every answer I found revealed 10 questions I had not known to ask. That is the nature of genuine inquiry. The question of what sound does to living matter, to the double helix that encodes the instructions for building a human being, is one of those questions that the more carefully you examine it, the more profound it becomes. The universe is not indifferent to vibration. The universe is, at its foundation, organized by vibration. The standing waves of quantum mechanics, the resonant frequencies of atomic orbitals, the phonon modes of molecular bonds. These are not metaphors. They are the physical structure of reality, as the mathematics describes it. When sound, organized, specific mechanical vibration, meets the organized vibrational structure of a living cell, something happens. The question is what? And the answer matters enormously. What do you think sound frequency research could reveal? I am genuinely asking. The most important questions in the history of science were not answered by lone geniuses working in isolation. They were answered by communities of curious people who refused to stop asking. If this investigation into the intersection of physics, sound, and biology is one you find worth pursuing, leave your thoughts below. What frequency research have you encountered? What questions does this raise for you? Scientific progress has always depended on the accumulation of questions as much as the accumulation of answers. If you have stayed with me this long, I encourage you to subscribe to this channel. More questions like this one, questions that sit at the edge of what we know and what we are only beginning to understand, are being explored here regularly. The distinction between what is known and what is merely assumed to be known is one of the most important distinctions a thinking person can cultivate. I spent my life trying to maintain that distinction. I did not always succeed, but I never stopped trying. My name is Albert Einstein, and I remain convinced, even now, that the most beautiful thing we can experience is the mysterious. It is the source of all true art and science. The person who can no longer pause to wonder, who can no longer stand wrapped in awe, is as good as dead. Their eyes are closed. Sound moves through matter. Matter responds. Life is more organized than we imagined, and more fragile, and more extraordinary. Keep asking the question.